investigator of the Howard Hughes Medical Institute. Now I'll turn it over to Augustus to talk about his education.
All right, so he completed his MD and PhD degrees both at the University of Chicago. From there, his research is focused on patterns of neural stem cell division, cell fate choices, and cell migrations in the developing cerebral cortex. Dr. Walsh has pioneered the analysis of human genetic diseases that disrupt the cerebral cortex.
He has also been recognized with the Jacob Javits Neuroscience Investigator Award, a top award of the NIH's National Institute of Neurological Disorders and Stroke, as well as the Derek Denny Brown Award, which is the most prestigious award from the American Neurological Association for outstanding contributions to scientific achievement.
Thank you very much. Should I take it from here?
Yes, please, Dr. Walsh.
Okay, great. Well, thanks everybody. It's a real pleasure to talk to a bunch of budding young scientists. We have two daughters who actually both went into various kinds of science, and I always enjoy having high school students work in our lab in the summer to just, it was very important to them to get an early exposure to science. I think it showed them that science is kind of a fun thing to do rather than the way it's often portrayed on TV and in the movies. So anyway, like I say, it's great to meet you all. So I'll tell you a little bit about my background. I went to college with no idea what I wanted to do at all. And I went to Bucknell University, which probably many of you have not heard of. It's a small liberal arts college in the middle of Pennsylvania. I grew up in a New York suburb. My parents are both New Yorkers. And I went to college after just 3 years of high school, not actually wanting to be in college or to go to college yet, but actually wanting to be a senior in high school. And so I really wasn't thinking about college when I got there. And I had teachers there who were very influential on me. The very first semester, I took a psychology course with Alan Leshner, who just really inspired me about how amazing the brain is and how it's just at the root of everything we do. And then I did other research with Owen Floody. I also had an amazing organic chemistry professor my first semester. And so that inspired me to take a sort of a chemical approach to try to understand the brain. And so ever since then, I've been fascinated by this question of how a bunch of undifferentiated cells in this one particular part of your body somehow become the organ of conscious thought and creativity and intelligence. And what is it that's so special about these cells, and how do they get that way? And so the brain has basically two different fundamental types of cells, excitatory ones and inhibitory cells. Excitatory ones make other neurons more likely to fire, and inhibitory ones inhibit the activity of their neighboring neurons. And so I spent a lot of my career trying to figure out how these cells develop in the hopes that that would somehow tell me how this, spark of creativity starts from these neurons in the brain. So the neurons in the brain are formed from undifferentiated stem cells during prenatal development. This is a schematic of a cross-section of a human fetal brain. This is what the cells look like. These red cells are the stem cells. They have these very elongated processes, and they're dividing cells. And they generate the neurons, which are shown up here. And can people see my pointer when I use it? Maybe, yes, great. I see Maggie nodding, so thanks for that. And so neurons are non-dividing cells, and so they actually are not in the cell cycle. So every neuron in your brain has what we call a birth date. It's the day at which that cell is formed from these dividing stem cells and becomes permanently postmitotic for the rest of its life. So neurons in the deepest layers of your cerebral cortex, layer 6, are formed first about 7 weeks after conception, shown in green here. And then the progressively more superficial layers are actually formed later and then have to migrate past these earlier born neurons. Wanting to understand how the brain gets there, I moved to do an MD PhD at the University of Chicago. And actually, unlike many of you, I'd never heard of an MD PhD program when I was in high school or even throughout college. I didn't even learn about MD PhD programs until I was like entering my senior year of college. And I'd worked a little bit in hospitals in college, and I wasn't really sure that being a doctor was for me because I found that to be a little bit boring. Uh, and then I worked in research labs, and I really loved doing that. Uh, and and then when I learned about that there was actually such a thing that you could do an MD and a PhD, and that they would actually pay your way to do it, so it was free. It was like a moment that I realized this was exactly what I wanted to do. And coming from a big family where we didn't have a lot of money, it also was really important. Everybody, I'm one of 8 children, and all of us got some kind of scholarships or something like that to go to college and graduate school. I did my PhD there with Ray Guillory, and he turned me on to the way genetics regulates the development of the brain. Although this was such a long time ago, this was back in the 80s. We knew very little about the relationship of genetics in the brain. But one of the first insights was his work that showed that people with albinism, or a lack of pigment, and in fact, all species of mammals that have a lack of pigment have abnormal connections between the eye and the brain. And that's why Siamese cats are cross-eyed, and that's why folks with albinism are cross-eyed. That's why even this Siamese tiger from the Chicago Zoo is cross-eyed. It's because the eyes don't project properly onto the brain. Ordinarily, some of a given eye, in this case the left eye, some of the fibers from that eye project to the opposite side of the brain, shown in blue, and some project to the same side of the brain. But in the absence of pigment, almost all of the fibers project to the opposite side of the brain. And this causes a completely disordered mapping of the world onto the brain. So that was amazing to me, how genes could regulate that process. But then what's even more amazing is that people with albinism have normal cognition. They can learn to read. And in fact, somehow the brain figures it out and rewires itself in a way that would not be normal for people with normal visual projections, but which is compensatory for their abnormal visual projections. And so this is even bigger mystery, is how the brain, when faced with this abnormal genetic inheritance, can actually, is so plastic that it can actually rewire itself so that it is functional. And so these are really 2 issues. One is, how do genes define the brain? And then also, how do genes not define the brain? How do the genes set up a process that allows the brain to be constantly changing its wiring in response to stimuli? And that's essentially the basis of how we learn things. Ray Guillory also introduced me to all the amazing characters that have done neuroscience over the years. The most inspiring of whom was Santiago Ramon y Cajal, who is often attributed with being the founder of modern neuroscience. And he was not only an amazing neuroscientist, but also a spectacular artist. So these are some of his drawings of neurons in the brain that actually, they could pass as fine art. They're just so beautiful to look at. And he was also a very inspiring writer. So here's one of his quotes. He described himself as being like an entomologist in pursuit of the mysterious butterflies of the soul, these things called neurons, that have these delicate shapes and elegant forms, but that somehow underlie the secret of our mental life. So after I decided during the MD PhD program, I wanted to stay in the study of the brain. And so I decided I was going to specialize in neurology, which is the clinical practice of taking care of people with disorders of the brain. And for my training there, I moved here to Boston, to Massachusetts General Hospital. And the reason I did that was because at that time, and still, that the Department of Neurology there was pioneering the use of genetics to understand the brain. And that was led by Joseph Martin, who was the chairman of neurology at that time. And he was working with Nancy Wexler, who did this amazing study of this huge family of patients with a rare genetic disease called Huntington's disease. Because back then, without a full map and a whole sequence of the genome, you could only study genetic conditions where you could find a large family, because we had only rudimentary ways of mapping genes in the genome. And so Nancy had identified this very large family in Colombia, but I wanted to study actually genes that regulated the development of the cerebral cortex, because I really wanted to understand how the mind gets that way. And so this was an example of one of those conditions where the normal cerebral cortex is basically turned into a double cortex. There's a layer of gray matter, and then there's a layer of white matter, and then there's a second layer of gray matter where it's not supposed to be. But these sorts of disorders, you would never see a large family of it because it causes severe intellectual disability and intractable epilepsy, so that the affected patients would never have a family themselves. And so I wanted to learn about genetics. I didn't really know how I was going to apply it to study this question that I wanted to study in my career. And so I continued to really do studies that were more descriptive of just how the brain gets that way. And to do that, I did a postdoctoral fellowship with Connie Sepko. And Connie had developed ways of studying clones in the brain. So she developed retroviruses, which carry a histochemical marker that turns the cells blue after you section the brain. And so this is a picture of a clone of neurons in the retina. Here's what the retina looks like. That picture of Cajal shows what the retina looks like with these layers of cells. And Connie figured out a way of labeling the stem cells. And so you would label a clone of neurons in the retina, and this is what it looks like. Basically, they're very tight packed clusters. They're so tightly packed, you can't tell where one ends and one begins because they're almost overlapping. And so I wanted to apply that to understanding the cerebral cortex. And what I found was that the clones on the cerebral cortex were not like the ones in the retina at all, because in fact, they scatter a lot. Here's a handful of cells derived from a common stem cell, but they seem to be migrating through the brain and going in different directions. This one seems to be going a little to the left, and these seem to be going a little to the right. And so it was very different than the retina, but that made it very hard to interpret. And so we needed a better tool. And so instead of using one retrovirus, I developed a way of making hundreds of different retroviruses, but we don't have hundreds of different colors. And so we used DNA as a way to label each clone with a different tag of DNA. I'd put random stretches of genomic DNA into the retroviruses so that we had 100 or 1,000 or even 1,000,000 different retroviruses with different DNA tags. And we could then label the clones and then cut out the cells one at a time, which was a little bit of a drag, but that's what you do when you're a postdoc. And then analyze these DNA tags to find all of the cells in one clone because they have one tag, and all of the cells in another clone because they have a different tag. And it turns out, in fact, that sometimes the clones would cluster like this blue clone, which is sort of like a pizza slice, going down from here, the ventricle where the stem cells are, up to the cortex where the neurons migrate to. And sometimes the clones were sort of coherent, although not by any means close neighbors the way they are in the retina. But sometimes clones would do the exact opposite. And so these are two clones that are shown in a side view of the surface of the cortex. And the blue one and the green one, they both scatter all the way from one end of the cortex to another, as much as they possibly could. And we later came to learn that the clustered clones were the excitatory neurons, and the scattered clones were these inhibitory neurons that actually were formed in a deep place in the cortex and would migrate far and wide. And so that shows how you can really, there's certain things about the brain you can only learn if you develop a new technology for studying it. And without this technology of tagging clones, wherever they would go, we could never tell exactly the extent of this dispersion of clonally related cells. But, and so I thought I was just going to basically keep describing how the brain develops until one day in May of 1993, just about 30 years ago, when I just had started my own lab in January of that same year, I went to a meeting in Venice with my wife, Mingwei Chen, who's a cardiologist. I'd met her as residents at Mass General Hospital. And I heard one of my teachers from the University of Chicago, a child neurologist by the name of Peter Huttenlocher, present this family with an inherited disorder that causes a abnormal development of the stem cells in the brain. And so what happens in this particular condition is that neurons are formed normally, but a significant fraction of the neurons never migrate. They're stuck along the ventricle, as shown in these yellow blobs, where the stem cells normally are during development. But in fact, the neurons normally migrate away from that ventricular surface, that deep channel, and they usually migrate out to the cortex. But in this case, many of the neurons migrate normally, but a whole bunch of other ones never migrate at all. They just remain as these abnormal blobs along the lining. And so this moment told me that I could actually use genetics, that we had learned enough genetics now to be able to start studying developmental abnormalities of the human cortex. And so we studied that condition. I also actually went back to study that condition that had inspired me during residency, that double cortex syndrome. And this condition, again, we found a couple of small families. Here's a mother who had 3 affected children. And we found that the double cortex syndrome is almost always seen in females, because males that have the same mutation have a much more severe condition where the entire brain is malformed and smooth instead of normally folded. And that's because this particular gene, like that filament A gene also, these are both genes on the X chromosome. And so the females are a mosaic of mutant cells and mutant cells and normal cells. In the case of double cortex, there are lucky neurons that shut off their X chromosome and use they shut off their mutant X chromosome. They use their normal X chromosome. So those neurons behave normally and they migrate to the normal place that the cortex forms, which is right in the outer surface of the brain. And then this band of abnormal neurons, this second cortex, is actually formed from unlucky neurons that shut off their good X chromosome and they express the genes on their abnormal X chromosome. And so they lack this gene double cortin that's necessary for them to migrate all the way to the cortex. And so they form this abnormal band. And so then over the years, we studied dozens of different genetic disorders that disrupt the development of the brain in all different ways. And we identified dozens of different genes responsible for these things. And of course, over the decades, the tools have gotten much better. It's become now possible to define the genetic disorder in a single child because we know so much about the normal sequence of the normal genome in normal people. And we realize that actually the genome, our genome is mutated so commonly that in fact the 8 billion people on Earth represent a family of mutations such that every single one of the 6 billion DNA nucleotides in our genome is actually mutated in somebody somewhere in the world. So there are 8 billion people on Earth, and we all have 80 mutations relative to our parents. In fact, every time the sperm and the egg are formed, there are dozens of new mutations present in the sperm and the egg that were absent in the parents. And so that means that every generation on Earth, there are 640 billion new mutations in our genome, which is actually big, but it's not that big. It's only 3 billion base pairs. And so that means that actually every site in our genome is mutated on the average 200 times just from one generation to the next. And so not only is every gene mutated, but in fact, every site at every genome is mutated in someone in the world. And so all of the genes that are essential for putting our brain together have been mutated somewhere among the people that are present in the world. They may not be observable because some of those mutations are lethal. And so the people or the embryos that carried them might have died, but in fact, they've all been mutated somewhere. So when you study something like the migration of neurons during cortical development, which normally, again, happens because these stem cells make these postmitotic neurons, and the neurons actually have to migrate a long way to get to the cortex. We can find disorders that affect all different stages of that migration. I showed you those patients that have blobs of neurons that never migrate, and that's actually true of several different genes that block a proportion of neurons from ever migrating. Or we have these conditions like double cortex syndrome. That's one of a half a dozen conditions where the neurons migrate halfway to the cortex and then stop in the middle of nowhere. Or there's another half a dozen conditions where the neurons actually migrate all the way to the cortex, but then when they get there, they go to exactly the wrong layer, so that the neurons that are destined for layer 6 end up at the top instead of at the bottom. The neurons that are destined for layer 2 end up at the bottom instead of at the top. And so the neurons are able to migrate okay, but they're targeted in a way that's abnormal. Or there are conditions where, in fact, the neurons migrate too far. They migrate right past the neurons, right past the cortex, and they burst out of the brain, and they form a tangled mass on the outside surface of the brain, which is completely non-functional. Or there are genes that disrupt the stem cells themselves, and so they result in a cortex which is smaller than normal. So this is a condition called microcephaly, micro meaning small, and cephaly just referring to the cortex. And this is a three-dimensional reconstruction of a normal human brain, and then the brain of a human that has this condition called microcephaly, where the brain can be reduced to about a third of the normal size. And we've identified more than a dozen different genes that cause microcephaly, and many of them function together as a complex of proteins around the centrosome, which is the organizing structure of the mitotic spindle. And so these proteins then function again in the stem cells. And so the neurons that are formed are actually not necessarily abnormal. There just aren't enough of them. And this microcephaly complex actually is essential in these stem cells. This is a zoom in on the stem cells. Here are the nuclei of the stem cells that are undergoing cell division. And this microcephaly complex, which is highlighted here in red, is basically present along the ventricular lining itself, but then also it links this ventricular lining to the mitotic apparatus. So you ordinarily see the green and the red together in the normal brain. But then if you lack one of these microcephaly proteins, you see spots of red and green that are separated from one another because they're not linked together where they normally are. And amazingly, if you overexpress this protein complex, you can make the brain of a mouse too big. And so that's what we did here. And so it's this complex and the proteins that line the ventricle work together to regulate the size of the brain. And this seems to be a process that regulates the size of the brain not only during development, but also during evolution. Over the course of this work, we discovered that some of these microcephaly proteins have actually been the targets of the evolutionary selection on the lineage leading to humans. And they seem to be, at least in part, responsible for why our brain is bigger than the brain of our nearest neighbors, because one of these microcephaly genes called ASPM has been systematically mutated over the course of primate evolution. So this made me realize that you could actually use human genetics not only to study development, but also to study the evolution of the brain. And this led to the founding of a Center for Human Brain Evolution, along with another geneticist, David Reich. And his specialty is digging up old bones of Neanderthals and ancient humans and figuring out ways of sequencing it. And so his work allows us to identify the parts of the genome that are particularly different between humans and non-humans. And then our work allows us to try to figure out which of those thousands of spots in the genome that differ between humans and non-humans actually have essential functions in the brain by identifying patients that have mutations in those evolutionarily dynamic parts of the genome. And then a neuroscientist, Mike Greenberg, uses molecular neurobiology to try to figure out what are those evolutionarily dynamic sequences actually doing in neurons? How do they relate to the process of cognition? So in the last 10 minutes, I want to talk about not the genetic diversity of humans on our planet, but in fact, the genetic diversity that lies within all of us. So it turns out that as genetically diverse as humans are across the world, each one of you has within you greater genetic diversity than all of the humans in the world put together. Because every cell in our body has a genome that's unique from that of its neighboring cells. And this is caused by the fact that every time a cell divides, it has to replicate its genome. And the copy is never as good as the original. Every time a cell replicates its genome, it creates a handful of errors. These might be a single substitution of one base pair for another, like a substitution of a T to a G or a C to an A, or it could be a larger structural change where a large chunk of a chromosome might be gained or lost. And so this can create mutations. And if they occur early in development, that mutation will be present all throughout the body, but only in a fraction of the cells of the body. Or there might be other mutations that occur later in development after the brain tissue separates from the non-brain tissue. And that would create a mutation like this yellow mutation. It's present all through the brain, but is absent in the lungs and other tissues because it happened after those tissues separated from one another. Or there are mutations that can be present, like this green mutation in one half of the brain and not the other half, or in like this blue mutation only in a quarter of the brain, or only maybe even in a little tiny part of the brain. And so these mutations are constantly occurring during development, but I'll show you that they even occur after the neurons stop dividing. And so they create a mosaic brain where every cell has its own genome, sort of like snowflakes, where no two neuronal genomes are the same. And this turns out to be really important for all kinds of diseases, as well as for the normal development of the brain. So we got started on this work by studying children that had intractable epilepsy, where the epilepsy was so bad that neurosurgeons would remove the part of the brain that looked abnormal. They'd sometimes remove an entire half of the brain, or they would remove a little pizza slice of the brain, like this little bright spot. And we discovered that these disorders are caused by mutations that we normally associate with cancer. They're some of the same mutations that drive cancer, but they occur in neurons. And so neurons don't divide, so they can't make cancer. But when neurons get cancer mutations, they don't cause cancer. They instead make the neurons intensely excitable, and that creates epilepsy. And so these are lesions. These are disorders that are stable. They don't grow. They don't make cancer. They do show a little bit of overgrowth relative to normal, but it's a stable overgrowth. And then the size of the lesion reflects, in fact, when during development that mutation occurs, like I showed you in the previous slide. So these large lesions, these large disorders have the mutation in about a quarter or the third of the cells throughout an entire half of the brain. And these smaller lesions have the mutation in only about 1% of the cells in a tiny area. The particular mutations aren't important. A lot of them activate a pathway called mTOR, but that's allowed us to repurpose some of the drugs that were developed for cancer to use them to treat children with epilepsy that we can't treat with surgery. And so it turns out that mosaic mutations are not only important for pediatric epilepsy, they're very important, we think, for adult epilepsy, but I don't have time to tell you about that. We think that they cause a portion of autism spectrum disorders, probably especially the higher functioning autism, where it seems like part of the brain works fine and part of the brain works very abnormally. We think they're also important in schizophrenia and in Alzheimer's disease, where in fact it looks like these cancer driver mutations occur in a particular type of glial cell. But I'll just give you one example of why they're important for the purposes of our talk today. And we got to understand them by actually developing ways of sequencing, again, a technology development, where we developed a way of sequencing the genome of just one cell. And so we would take the neurons, of the 80 billion neurons from a human brain, would take a little patch of brain, would separate each nuclei. That's these dots are individual neuronal nuclei, and then we'd take a couple of nuclei and put them into a plate and amplify the genome and sequence it. And you can compare the sequence of one neuron to the sequence of another neuron, the way you would, using the same tools we would use to compare my genome to the genome of one of you. And just like our genomes differ from one another as people, turns out the genomes of neurons differ quite a lot from one another. And over the years, we've discovered transposon insertions or large gains or losses of parts of chromosomes, or single nucleotide variations, or small insertions or deletions. And so I'll talk about large-scale gains and losses of whole chromosomes, which turn out to be surprisingly and kind of scarily common during the early development of the embryo. So here's the results of a study from a group in China from a couple of years ago, where they took the early embryo, the blastocyst, and they separated out single cells and compared their sequence. And some of the cells, and this is what it looks like. This is the chromosomes of a single cell. Here's chromosome 1, chromosome 2, chromosome 3, and this shows, the blue dots show that basically all of the chromosomes are present as 2 copies. That's what's supposed to happen. But then here's a cell that has, instead of 2 copies of chromosome 16, has 3 copies, highlighted in red. Or here is a different cell that's missing a copy of chromosome 19. It's also missing an X chromosome. And so this turns out to be really common. In 20 different blastocysts that they studied, all of them had cells that had gained or lost a chromosome, typically about a quarter of the cells, shown here in red. And so we wanted to know what happens to the neurons in the brain. Do they maintain these large-scale gains or losses of chromosomes, or does it change over development? And there had been early suggestions that maybe neurons had lots of gains or losses of chromosomes, but in fact, more recent studies showed that that's rare. Here's an example from mouse, from human, that suggests that aneuploidy is present in only about 5% of the neurons in the adult brain. And so how does this change from 25% of the cells in the early blastocyst to only 5% in the adult? And so Diane Chow, who's a child neurologist working in my lab, developed a method where we could sequence thousands of neurons and look at this at high high throughput. And I won't talk about the details of it, which aren't important, but this is what the readout looks like when we look at neurons from prenatal human brains, shown on the left, and compare it to neurons from a postnatal human brains, shown on the right. And again, the chromosomes are arrayed around the clock now. Here's chromosome 1, chromosome 2, chromosome 3, chromosome 4. And then gains of chromosomes or gains of half of a chromosome are shown with red shading, and losses are shown in green. And what you can see is that the prenatal neurons have lots of gains and losses, but the postnatal neurons have far fewer. And so somehow neurons have become less abnormal. How does that happen? And so we sorted the neurons into those that are really abnormal, where they're missing lots of segments of chromosomes, multiple places in the genome, or ones that are a little less abnormal, where there's losses of small segments, or maybe neurons that have lots of gains but relatively fewer losses, or neurons that look pretty good. And it's particularly these very abnormal neurons in these first 2 categories, the blue categories, that are particularly common, affecting about 40% of the neurons before birth, but affecting fewer than 20% of them after birth. So about half of the neurons with these very severe genomes are eliminated. They go away. How does that happen? Do the neurons, the genomes fix themselves, or are the neurons containing these abnormal genomes killed off? It turns out we think they're killed off. We know that in the developing brain, there's a wave of death of neurons. This is shown here in the mouse, and you can see it. These are cross sections of a part of the brain called the hippocampus, stained with a dye that shows dying cells as black. And you see that there are very few dying cells in early embryonic development, but then a few days later, you can see neurons starting to die, and there's a wave of cell death. And then by the time the mice are born, here is postnatal day 5, there's almost none of this black staining anymore. And so there's a wave of death of neurons. And that's been known for a long time. And it's thought that this wave of death in humans actually occurs in the second half of gestation, after about 25 weeks of development. And this death of neurons has long been known to relate to the ability of neurons to form connections. Neurons normally form connections with one another. These connections are called synapses, and that's necessary for neurons to communicate. And if neurons fail to make synapses, they die. And that's actually been known for 150 years. It was discovered by this guy, Professor von Guden. He studied people who had lost one eye and then survived for several years before dying. And what he discovered was, here's a picture from his publication from 1890. He discovered that people lost one eye and then died a couple of years later. Parts of the brain that receive inputs from that lost eye are shrunken. In this case, the larger projection from that lost eye is to the opposite side of the brain, and this little body called the lateral geniculate nucleus is shrunken in this person compared to the normal size. And in fact, that the part of the brain that receives input from the eye has layers. Each layer receives input either from one eye or the other. And you can see that if you section that part of the brain after someone has died after losing an eye, it's specifically the layers of the brain that receive from the lost eye that are shrunken. And so when you take out the eye, of course the eye dies, but also the parts of the brain that connect to the eye die, and even actually some of the neurons that are a whole other synapse away die. So neurons then are social cells. They have to form connections in order to survive. And so we wanted to know whether there were particular parts of the genome that were essential for the neurons to survive. And for that, we used this analysis that was done on airplanes that came back from bombing missions during World War II. And there would be planes that would successfully return to base with bullet holes, and that would identify the places where you could make a bullet hole in a plane and it could still fly and still survive. So these are called hotspots. But then there are cold spots where none of the planes returning to base would ever have bullet holes, and that's because they're absolutely essential. If there's a bullet hole in the cockpit, that's going to kill the pilot, so the plane will not be able to return. And so those cold spots, we looked for cold spots in the genome that were never deleted in the neurons that survived, and those cold spots contained a lot of genes, as you might expect. But they not only contain just any genes, they contain genes implicated in causing autism spectrum disorders, or genes that were essential to make synapses. And so this suggests that the neurons are formed, but then they go through a competitive process of forming synapses, and the neurons with very abnormal genomes die off because they can't form synapses normally. And so there's essentially a Darwinian selection of neurons after they're formed to preserve those that have relatively normal genomes and to kill those that have abnormal genomes. And so the neuron, the brain somehow has this ability to interpret the quality of a genome and kill off cells that have abnormal genomes. And this dying then is particularly dramatic near the end of gestation, between the end of gestation and birth, but actually it continues throughout life. You can see that newborns have a larger number of copy number variants, so gains or losses of pieces of chromosome, than in fact aged people do. So some of you actually have neurons that still have copy number variants that are going to be killed off sometime in the next few days or weeks or years. And this has all kinds of implications because it suggests that to a certain extent, the process of learning is a process of elimination. It's a process of death that kills off neurons that are less than fully functional. And anything that disrupts this process of Darwinian selection would leave our brain with impaired neurons. If there might be a difficult birth, a premature birth, maybe abnormal nutrition, maybe germline genes that are associated with intellectual disabilities or autism, we don't know yet, but they might actually modify this process of competitive selection. And finally, then I'll tell you that this elimination of imperfect genomes is not necessarily a good thing, because our genomes are becoming more abnormal as we age. And so we discovered, when we started sequencing these genomes of single neurons, that that unlike these large-scale changes, these copy number variants, these get better and get less common with age, but other kinds of mutations become more common with age. Substitutions of one DNA base, like a C to a T or a G to an A, these actually are relatively uncommon in neurons when we're first born, but increase by about 20 per year. And that has been shown now with 5 different methods. It's a fundamental, seems like a fundamental thing, that we get about 17 or 18 of these single nucleotide substitutions every year of our life. 17 the first year of life, 17 the 90th year of life. And then we get about 2 or 3 of these other kinds of mutations, which are short insertions or deletions. And so in total, about 20 of these mutations. And neurons actually accumulate these mutations faster than hematopoietic stem cells, but slower than other tissues. So all of our tissues accumulate mutations like this, but the surprising thing is that neurons accumulate them as fast as other tissues, even though neurons are non-dividing cells. Unfortunately, while the copy number variants then decrease with age, these other kinds of mutations increase with age, and they get a lot more common in degenerative diseases. And so this process where the brain is constantly searching for a perfect genome and eliminating the genomes that are imperfect, it works great during development by eliminating the genomes that might be a problem for learning and memory, but then in fact it becomes self-defeating. This same process carries the seeds of destruction of neurons as we age, because as we age, these other kinds of mutations can become so common that if they engage the same mechanism, they can cause a potentially widespread death of neurons. So in summary then, the genes are, the brain is formed by genes that define an initial set of programs that make neurons in the right numbers and put them in the right places. And then there are different programs that actually regulate this competition between neurons and that dictate changes in synapses and that eliminate neurons that actually are not fit. And so that's synaptic plasticity is a lifelong competition for survival between neurons. And that each neuron's unique and changing genomes seems to define its success in that competition. So then I'll just end by mentioning saying that everything I told you about was not done by me, but was done by all of these amazing students and postdocs that have come through my lab over the many years that we've had a lab. And it was all inspired by mentors that I've had along the way, whether it was back in college or during graduate school or during my postdoctoral training. So thanks very much. We have plenty of time if people want to ask questions, and maybe I can stop showing slides and stop sharing my screen unless people have a particular question that causes me to do that.
Thank you so much, Dr. Walsh. Augustus, Azzy, I'll leave it to you guys to field questions. And at the end, if we have a little more time, I have a question myself.
Awesome. Augustus, do you want to ask a question first before we hand it up to the crowd?
Yeah, I can ask a quick question that I had. So something that I've had throughout my life is Tourette syndrome. I was kind of wondering if, talking about the epilepsy that you talked about with the clusters of excitable neurotransmitters or whatnot, have you looked at that at all? And do you know if that could be related to that?
That's a fascinating question. It's such a complex syndrome. And the same sort of thing. It looks like it involves maybe just one part of the brain, but definitely not the whole brain, because most folks with Tourette's, are plenty smart, just like you are. We have not looked at Tourette syndrome because the sorts of experiments I'm describing can only be done after someone dies and we have their brain at autopsy. There is a group at Yale that's been investigating the same hypothesis that there might be mutations present in part of the brain but not others. They haven't yet discovered that that is the case, and so we don't know yet one way or the other whether that's the case. But I tell you, I'm very interested in that. I think that it's a fascinating possibility, but it just makes so much sense that that disorders like this, where they're, like I say, where people are perfectly normal, except for just one little thing. And that's of course true of all of us, right? We all have our strengths and challenges, and we all, in fact, have clones of neurons that are not normal. And so we don't know really, broadly speaking, to what extent clonal abnormalities in the brain might shape the personality of all of us. So thanks for that question. It's a great
Awesome.
question.
Thank you.
Before we give it to the crowd, I would like to ask a question too, and I think many researchers would be really interested in hearing this, especially in high school. Your research, Dr. Walsh, is absolutely amazing, but it also seems like there were several iterations and a lot of people involved before you came to your conclusions, and it seems like it took a lot of patience. So when developments take a long time or discoveries might be uncertain, how do you stay motivated, organized, and focused, and what advice would you give to high schoolers who want to follow your path?
Well, gee, that's a great question, Azzy, and you're right. I mean, so one thing I like to emphasize is that it's easy, like when I was your age and looking up at professors or something like that, I would think they were really different than me. That they sort of had it all figured out. I was like a completely disorganized and not always, my motivation would sort of come and go. I was doing a lot of crazy stuff as a younger person. And I always just thought that, the people who were really successful were like fundamentally different, that they always kind of had like this, the sunshine was always like right over their shoulder, pointing them in the right direction. And here I was just kind of like, groping to try to figure out what to do. So I'm just here to tell you that that's what I was. I was like, I had no idea what I wanted to do when I went to college. I entered as an undeclared major. And I think, like I said, the MD PhD seemed like that seemed like an easy decision for me. It's not an easy decision for everybody, but it was like a moment where it just seemed to crystallize. However, I had no clue what I was actually going to do with that and how I was going to get there. How I was actually going to do like clinical medicine and science at the same time, and what was I going to work on. And so I just kind of kept, and even as late as after I'd finished my neurology residency, and when I was a postdoc with Connie doing those studies with our retroviruses, I still had no clue really. That made no sense with being a neurologist. It was very basic studies of mice and rats, uh, and so then it started to make sense when I heard about that family and I started doing genetics. And so I would say basically my advice would be try different stuff, uh, try to do things that you like, but also I found it useful to do things that I'm good at. Like I'm not good at math, for example. I'm the worst mathematician in my family. My wife and both daughters are better mathematicians than I am, like by a long shot. And but but anyway, things that I had some success with, I tended to stick with. And I think that's good because it's good to feel good about yourself. But I think you just have to try different stuff. Science will definitely be right for some of you people, but not all of you. Uh, and and then just kind of do things that you like, even if it's not what other people think is the right thing for you to do. Other people thought, I should do medicine. I don't actually see patients now. I am head of a clinical division, but don't see patients. I loved it when I saw patients, but I like even more the work in the lab. And so it's just hard to know, what's going to be right for you until you just give stuff a try. And I think then also you just have to be okay with the fact that it's going to work itself out, that you don't have to know today what you're going to be doing for the rest of your life. And in fact, your career changes every couple of years anyway. Your fate is not a fixed thing. Like even when you're a professor, the first couple of years, you're really focusing on your lab. The next couple of years, you're focusing on more teaching and recruiting people to your lab and growing your lab. The next couple of years, I'm really focused on the success of my students and my trainees. And a lot of what we do in the lab is focused on making them successful, and it's because of their good ideas. And I basically just kind of try to enable that.
Thank you so much. Those are some really good insights, Dr. Walsh. Now I'm going to start calling on people in order that they show up, and then me and Augustus will be taking turns picking on people. So first we have Maggie. Do you have a question?
Yes, I do. So, Dr. Walsh, that was an amazing talk. I really enjoyed it. You were talking about how some neurons fail to migrate toward the external parts of the brain, or even halfway, or even maybe go too far. What does that look like in the form of lifestyle of the patient?
So that's a great question. And it's completely variable from one condition to the next. The condition that I first started on, where there was those blobs of neurons that never migrate at all, you'd think that would be really bad for you. But in fact, it's not. That's why there was that big family that Peter found. Basically, those patients, their problem is epilepsy. It typically starts when you're a teenager, not as a newborn. The average IQ of those patients is about 10 points lower than normal, but you can be incredibly smart with that condition. You can be I've seen patients that are CEOs or medical students or computer programmers. And so it's a wide variability. And then, in fact, not even everybody has epilepsy. About a third of people have no epilepsy. And some people are asymptomatic. They're only discovered by accident when they have an MRI scan because they have a headache or something. But other conditions like the double cortex syndrome, that's much more severe. Even though the neurons are closer to the cortex, in fact, somehow worse. And we think it's because the neurons are very abnormal in their shape and their structure, in addition to being in the wrong place. Whereas the neurons that never migrate actually look pretty normal in their structure. And in fact, we think that the neurons that are forming those blobs and never migrate, we think they actually wire up to the normal cortex. And those people then are thinking with a brain that looks totally different than yours and mine. And yet, their mind is indistinguishable from ours. And so it's really variable from condition to condition. Most of the genetic conditions that disrupt the development of the brain cause severe intellectual disabilities and typically seizures. But as I say, some of them can be milder.
That was a pretty good answer. Before we continue asking questions, Dr. Walsh, we kind of wanted to know how much time do you have, or how many questions do you feel comfortable answering?
I can go a little bit. I'd like to answer as many, as many questions. I'll try to give shorter answers instead of prattling on so long. And I can go a little bit past the top of the hour, at least until 5 after, maybe a little longer. So hopefully we can get through the people that have their hands raised. And I'll just answer briefly a question in the chat from Aditi Arun. Because I think some of the other people might want to know the answer too. He or she was asking about, when I did the MD PhD, did I have to pay for anything? And basically, I did not have to pay for anything. The MD PhD program is supported by grants from the NIH, although NIH has canceled Harvard's MD PhD grant. And that particular grant allows students to come right out of college, and your tuition and all of your fees are paid by the grant, and you're paid a modest stipend. Might not be enough to live on in Boston, but it's at least something. And so I came out of medical school with very little debt. Now, that is the NIH-funded MD PhD programs. Most MD PhD programs are funded by NIH. Although in order to be supported by that, you have to be a US citizen or a green card holder. There are other MD PhD programs at some schools where they don't happen to have the NIH funding, but they may still allow you to do the program. So there are some circumstances in which people will do the 2 degrees and they'll pay their own way through medical school. But pretty much any PhD program in the biomedical sciences is not going to cost you money. It's going to be funded by some sort of grant or else funded by the institution.
All right. I'm going to call on Leon next.
Thank you. Hey, Dr. Walsh. I want to first thank you for taking time to speak with us and answering our questions. My question for you is, are some of these genetic mutations actually beneficial for your brain health?
So that's a fantastic question, Leon, and we don't know whether this genomic diversity that the brain originally has, and then whether that might be beneficial. I would love to study Einstein's brain, which is in a museum in Philadelphia, to see if he had some special clone, with some special clone in a particular gene. It's quite possible. Again, we all do have clones of cells in our brain that have mutations that alter the function of a gene. And it might be very important for, say, how identical twins are mostly identical, but not completely identical. And so you can imagine how it can be evolutionarily useful to create diversity and then select for diversity that still is consistent with making good synapses. And so I suspect that there might be a role for that. It's a little hard to investigate scientifically because it's not a black and white question of what's good and what's bad. And science prefers black and white questions. We're trying to figure out a way of getting at that, Leon.
Okay, thank you.
Yeah, thank you.
That's really interesting. I'll be calling on the next person, but after this, I'll have to get off the call for a lecture. So Augustus, you'll be choosing people for the Q&A for the rest of the time. But next up, we have Suchir. Do you have a question?
Yeah, I do. Thank you so much for the presentation, Dr. Walsh. This is kind of building on Azzy's question, but I'm wondering how young researchers today can pursue ambitious and long horizon ideas in a research system that increasingly prioritizes short-term validation.
Well, I mean, I think we don't really know what's going to happen in the present situation, right? I mean, the big unknown is whether the NIH budget is going to get cut as much as President Trump wants to cut it by 40%, and whether the National Science Foundation is going to get cut as much as he wants. I was reading an article this morning that suggests that in the case of NSF, it's probably not going to be cut anywhere near as much as he wants it to be. And the same, we're optimistic the same might be true of NIH. And so, I have been funded by the NIH for decades, and I've been very fortunate to be funded by the Howard Hughes Medical Institute as well. It gives me a lot more freedom to propose to do science which is totally out of the box. However, the NIH, before I was Hughes, I also had funding from the NIH that it does allow you to take risks. I think it's unfairly portrayed as being overly motivated by short-term gain and things like that. I think in order to be successful in science, you have to publish, but you can still the present biomedical program, if it's not completely destroyed by the present administration, is still really good at fostering plenty of scientific innovation. So I think if it's just not killed, it's going to continue to promote innovative people like, hopefully, people like you.
Thank you.
All right, next up, I see Ria.
Thank you. Thank you so much for the presentation. I found it really inspiring. And my question was about line 1 elements and retrotransposition in your paper, because your paper mentions retrotransposition activity contributing to somatic variation in the brain. How do you view the role of mobile genetic elements? Do they serve as noise, or could they be a hidden layer of regulatory plasticity?
Yeah, that's a great question, Ria. I tend to be more of their noise. So what we see is, multiple different types of diversity between one neuron and the next. Line retrotransposons being one of them, copy number variation being another, single nucleotide changes and small indels being others. And there's other types that we haven't had the technology yet to understand. Transposon mobilization was claimed by people to be very common, in which case, and that if it's common, then it might be a normal generator of diversity. Actually, we found that it's not that common. It only happens less than once per genome, and into sites that appear to be fairly random. And so we think it's a contributor, just one contributor of many to the variation of one neuron from another. And the other actually forms of variation are probably quantitatively at least more important.
Thank you.
Yeah.
All right, next I see Asha.
I actually have two questions. How do you think that the human and animal brains will like evolve over the next 1,000 years? And do you think that it'll be like the evolution will be driven more by genetic mutations, or do you think that technology will override biology in the future?
Well, golly, the second question, I don't really. Well, humans continue to evolve. I mean, the fact that we have 80 mutations compared to the last generation, what happened? The mutations just keep happening, and then evolution selects for them. I'm fascinated by the fact that, so what sort of selection pressures do humans face? Well, things like we have less strong selection against people that have immune disorders because we have ways of treating that. We have less strong selection actually against large heads. With a cesarean section being so common or in vitro fertilization, historically, our head could get to a certain size, but no bigger because it had to come out through the birth canal. That selection against really large heads actually is being removed, right? By in vitro and by the widespread use of cesarean section. I mean, cesarean section happens in 20 or 30% of births in some countries. And so I'll be curious to know a few generations from now what the average head size is, because that is a selection pressure. That's a balancing selection pressure. So the major selection in humans is generally infectious disease, right? I mean, the flu epidemic of 100 years ago killed something like 20% of the world's population. That's a significant selection pressure. And COVID didn't kill quite that many. And so we don't know what the selections are going to be in the future. But basically, in principle, humans will continue to evolve because mutations keep happening and we're always under some kind of selection. Although, again, with our improvement in lifespan, the selection pressures have changed.
And I have another question. You said that humans will keep on evolving and with mutations. Do you think that in the future that we'll be able to treat and detect more dangerous mutations earlier on and maybe like replace them with bioengineered clones of the cells?
Oh yeah, I mean we have the technology now in principle, in theory, to if you do in vitro, they'll harvest one of the blastocysts and do some sequencing on it to see if there are particular kinds of mutations. So in principle, that could be extended to look at the entire genome if you do in vitro fertilization. In principle, we could, sample, do early sampling of the embryo at the chorionic villus stage of everybody if we wanted to. It's just not advisable and it's too expensive. And so I'm sure that as technology develops, we will someday have the technology to look at the whole genome of people at an early stage of development. The ethical challenge will be what to do with that information.
Okay, thank you.
Alrighty. Thank you very much, Dr. Walsh. We want to be mindful of your time. It's already past 5. So
great.
Thank you. We really appreciate it. And yeah, thank you to all of you guys as well. Thank you, Augustus, Azzy, for moderating. And this was a lot of fun. Thank you
All right,
again,
sorry,
Dr.
that's
Walsh.
all the questions. Thanks very much.
Thank you, Dr. Walsh.
Bye, everybody.